So we're now a few minutes after 8 o'clock, so I think it would be a good time for us to get started. It gives me great pleasure today to introduce today's speaker, Dr. Greg Lowry. Greg is the Walter J. Blanco Senior Professor of Civil and Environmental Engineering at Carnegie Mellon University. He is the Deputy Director of the NSF EPA Center for Environmental Implications of Nanotechnology and an Executive Associate Editor of Environmental Science and Technology. His research aims to understand how the unique properties of engineered nanoparticles or nanomaterials affect their transport and fate in environmentally relevant matrices, including plants, soil, and groundwater. He uses this knowledge to help make crop agriculture, water treatment, and environmental remediation more sustainable. He's authored 194 peer-reviewed journal articles and 1 book. He's a board-certified environmental engineer, fellow of the American Association for the Advancement of Science, fellow of the IIES, and was a member of the National Academy of Science Committee on Science Breakthroughs 2030, a strategy for food and agricultural research. Dr. Lowry holds a BS, Bachelor of Science in Chemical Engineering from the University of California at Davis, a Master's in Science from the University of Wisconsin at Madison, and a PhD in Civil and Environmental Engineering from Stanford University. So welcome today, Greg.
Great, thanks, Doug, and thanks for the invitation to be part of your seminar series. I saw the last 2 names pop up, Kevin and Jason, I guess, for the last 2. So really, really good group. Let me go ahead and share. All right, we good on that?
Yep, that looks good.
Okay, great. Yeah, so I'm going to share a little bit. It's probably going to be some overlap with what Jason talked about, Jason White, but not a huge amount. I think Jason and I take different approaches to this, and he and I have known each other for a lot of years. And what we're trying to do is, rather than develop technologies, although some of the things I'm going to show you today are new technologies, we're trying to develop the understanding of how nanomaterials interact with plants, how they move around. Think of it like fate and transport. We used to do a lot of fate and transport of particles and chemicals in the subsurface and groundwater. Now we just have a different porous medium. It happens to be a plant, which turns out to be quite interesting, actually. So I always like to start with kind of the people who do the work. So, you know, Eleanor here on the left, she kind of got us started in all of this plant stuff. And we originally were working with roots and looking at how things went up in roots. And we do a lot of X-ray spectroscopy work to understand where things are and what their chemical speciation is. And then I had Astrid in my lab for, I guess I should say Eleanor's at Stanford now. I have Astrid came to my lab and she was with me for about 3 years and started working on foliar uptake and delivery. She's since taken a position back in France at CNRS. Elin actually just graduated last year and has been with me for the last 8 months or so, and is moving to MIT in August. But he started getting us thinking about polymers and how we can use polymers with well-controlled chemistries to move them around. And a lot of what I'm going to talk about today is actually is his work. But I've been fortunate to work with Enzo Lombi in Australia. He's been really great. A lot of this synchrotron mapping couldn't have been done without him. I sort of have this new crowd that I hang out with, Juan Pablo Giraldo and Nicole Steinmetz. Juan Pablo's at UC Riverside and Nicole's at UC San Diego. And we've been looking at how to leverage these delivery methods with our polymers and some other methods to try and deliver DNA inside of plants. And I have a whole new group of students over here that have been working and are working on these projects. So it's been really fun for the last 6 or 7 years working on this, and I'm sure there's many more to go. So why do we do this? Well, Jason probably showed something similar to this, but agriculture uses an awful lot of our resources. Right, it accounts for a lot of the greenhouse gas emissions that society produces, uses a lot of energy, a lot of land. Most of our fresh water goes to agriculture. Most of the deforestation that's going on is attributed to agricultural activities. So it touches on most of the sustainable development goals, and it uses a lot of resource. And of course, the world population continues to grow, and we're going to need to feed those people, and we're going to have to do so on less land, with less soil, and with lower impacts than we have been in the past. So this idea of sustainable intensification, I always, yeah, this word's been around for a long time, or this concept, but thinking about intensifying agriculture in a sustainable way is a daunting task. It's going to require an awful lot of us to be focused on this. So that motivates us. Another big motivator for us is how inefficient agrochemical delivery actually is. So, I don't know if you guys know the numbers on these, and I don't know people's backgrounds, but for nitrogen fertilizer, half at best of what's being applied to crops actually goes into the crops. Most of the excess runs off, and the picture on the right here is Lake Erie, showing an algal bloom, and most of that algal bloom is because of nitrogen runoff. And then, we're even less efficient with our pesticides. 10%, sometimes 1%, sometimes even less of what's applied to a crop actually makes it to its target and is running off in the environment and causing a sort of non-target or off-target problems. So we really need to fix this inefficiency problem. And then more recently, I guess I can just pull this up, is climate change. Climate change is threatening food in a way that I think is coming faster than anybody would have thought. It wasn't until very recently, I think this New York Times article came out right around the time I was on that National Academies Committee looking at the future of food and agriculture. And it finally came out that says that climate change is threatening our food supply. Up in the upper right here, this is California, where they say 90% of the vegetables and foods in the US are grown in this area. I'm not sure I believe that number, but there is a lot of food grown in this area, and you can see it's a massive drought and it's pretty much a desert. So that's problematic. Very recently, I don't know if you've seen these stories, but these heat waves in India are projected to lower wheat harvest by about 6%. So heat damage of crops is an important one, and in fact, one that we've been working on, and I'll show you some interesting data on that. And then, of course, with climate change comes increase in pests. So there's a locust outbreak, and then an increase in use of pesticides to manage that. So it's sort of this spiral of effects where we've got this climate change and we don't have the tools to manage it just yet. So we're going to use our old tools, which aren't the best tools and aren't so great for the environment. All right, so nanotechnology is kind of where this whole thing is focused, and where this seminar series seems to be focused. So nanotech does offer some solutions. It's not going to offer every solution, right? We're not going to solve the world's hunger problems with just nanotechnology, but it's one of the tools that we have that I think is going to make some difference. And I'll show you some technologies that I think will do that work for us. A number of things are proposed for agriculture. In fact, ACS Nano just had a recent editorial by some new editors they brought on that talks specifically about the role of nanotechnology in agriculture and what we do and do not know. But clearly we can do a better job with efficiency, delivering both macronutrients and micronutrients, and I'm sure Jason talked a little bit about delivering copper and some other things to plants. There's also this idea of plant protection. We can deliver pesticides and antibiotics better. I'll show you some data on delivering anti-stress agents, which help plants overcome some of the stress from climate. People are working hard on delivering DNA into plants for a number of different reasons. One, genetic modification of plants, which I think is a tool we're going to need moving forward, but also just delivering plasmids to plants so that they'll manufacture drugs, or they'll manufacture vaccines, or they can make all kinds of products for us. So delivering things into plants that way is a challenge. People talk about seed coatings and various fertilizers and playing around with the microbiota for nitrogen fixation. All of these require some form of precision delivery. And I think this is where some of the problem lies, is that we don't exactly know how to deliver these efficiently to where they need to be. All right, so that sort of brings us to where we're focused in our lab right now. We believe that there's opportunities for targeted delivery in plants. Think of it like drug delivery in humans, except that we're doing it in plants. And there's a lot of similarities between plants and animal cells, but there's a lot of differences. Mostly the cell wall is a difference and leads to some problems. But there's also different barriers to overcome. With humans, you just you make a particle and you put it into a solution and then you inject it intravenously. It's a little harder to do that in plants. People do that in trees, for example, but it's hard to imagine doing that to every wheat plant in a field. But there are opportunities for targeted delivery. So we are targeting the phloem. So I don't know how much people know about plants, but maybe I should just say a couple words about plant vasculature. So plants kind of have arteries and veins like animals do. There's something called the xylem system, which just think of it as the roots pulling up stuff from the soil and bringing it up into the plant. That's what the xylem does. But then there's the phloem, and the phloem is taking the sugars and things that the leaves make and then putting them back down into the roots and distributing to the rest of the plant, including the fruits. This phloem system is where a lot of disease occurs. The phloem system is also much bigger, much faster, pushes things a lot easier than the xylem does. So as I said, we started with roots, but we ended up moving to phloem because it's a better system to deliver things. So there's all kinds of reasons to want to deliver things into the phloem. So we do target that, and I'll show you some data on that. You may want to target roots. So there's a lot of pathogens that are in roots. Fungi, and Jason probably talked about fungi and some diseases, some root diseases. If you could imagine putting something on the leaves and then moving it down into the roots and having it available to manage those diseases, that good reason to target roots. Then things like the mesophyll, which the mesophyll is just all this cellular material inside of the leaf. So this is a leaf cross section here on the bottom right. And then, of course, the center of that cross-section is the vasculature, where the xylem and phloem bundles are. But you might want to put things just into those phloem areas, because a lot of bacteria and diseases are inside the phloem. Plus, it's these cells that get stressed when there's excess heat or light or drought. You may want to hit the chloroplasts. So the chloroplasts are these green things. They're what make the plants green. They're inside of the cells. So if you look at the mesophyll, inside the mesophyll, there's this chloroplast. You might want to deliver something there because that's where photosynthesis occurs. So if you're trying to promote photosynthesis, that's a good target. And then, of course, you might want to hit the cell nucleus, because the cell nucleus has a bunch of machinery there for protein expression. So if you're trying to manufacture things in plants, you'd want to put things into the cell nucleus. So bottom line is there's multiple targets to go after. And that's really what we're trying to do, is figure out how to deliver things to those different targets. Now, the problem is there's lots of barriers. So with plants, you can inject things into plants, and it's hard to hit the phloem bundles and the vasculature, but you can do it. More often than not, we just want to spray something onto the plant if we can, foliarly. But when you do that, if you look at this upper right here, you first have to get across this cuticle, which is this waxy layer that kind of is made to keep things out. And even if you can move through the cuticle, and things like silhouette help disrupt cuticle, so you can put things onto this epidermis. But then you have this epidermis layer, which is very similar to ours, like it's your skin, your outer layer of cells. Its job is to keep things out. So you've got to penetrate through that epidermis. And I'm showing you all this in detail. I'll show you some pictures later. So I want you to have this kind of picture in mind when you see the images a little bit later. But once we move through this epidermis, then we can move through these cells in the mesophyll. So you could either move through cells or you could move around cells. So it's called apoplastic and symplastic transport. We'll play around with that a little bit and try to understand which works best and what's happening in leaves. And ultimately, you want something that you put on the leaf to move into this vasculature and then move to the rest of the plant. Okay. But there's so many barriers, and I talked about all of them so far. The other barrier here is a cell wall. So what's interesting about plant cells is they have a cell wall that supports this entire structure. So you got to get through the cell wall. Once you get through the cell wall, then you got to get through the cell membrane. And once you get through the cell membrane, you can get inside the cells, but then there's another membrane in the chloroplast. So just all kinds of barriers to putting things where they need to be. So I think this is a scientific challenge that ought to keep us going for probably another 20 years. All right, so does this stuff actually work? This is one of my favorite examples back in 2018, very simple experiment. You put a plant under drought stress and you treat the plant, in this case with cerium dioxide particles, which are antioxidants. They scavenge reactive oxygen. And it turns out when you spray a plant, with the nanoceria, you actually get more grain yield than you would under drought compared to the controls. So what's happening is these particles are going onto the plant. They're somehow getting inside or interacting, and they're scavenging reactive oxygen, and they're helping the plant manage drought stress. And this came out. It wasn't clear exactly why this was working. It wasn't clear where things went, but it worked, and it kind of piqued our interest, and we said, hey, we could do something with this. So we've since kind of taken that a little bit further, and we thought about things like managing heat stress, and then managing stress in general by just scavenging ROS, a little bit like that cerium dioxide particle would do, except that our materials, when they scavenge the ROS, then ultimately release something. In this case, it's magnesium. The magnesium helps enhance photosynthesis, and you end up managing that photosynthetic stress a little bit better. And then on the heat side, we know that we talked about that problem in India. Heat's just not good for crops. You can see in the bottom left here, these are heat stressed, probably maize, and it just doesn't look good. And we know that the average daily temperatures are rising, and it's actually this little rise here, a couple degrees, is not what we're worried about. What we're worried about are these series of days where the temperature goes extreme, like 50 degrees in India. And if you have 50 degrees for an extended period of time, the plants just can't handle that. And it often doesn't last a long time. Maybe it's only one day. Maybe it's only a 12-hour period, because then nightfall comes and the temperature comes back down. But if the plant can't manage that one hot cycle, then it pretty much dies, and you lose the crop. So we're working on ways, and I'll show you some data here. We're working on ways to manage that heat stress in a way that keeps the plants alive, even if they're stressed pretty hard. All right, so the overall objectives of our group is to develop these approaches for targeted delivery inside plants. And we described some of those opportunities a few years back. We're trying to develop these, what we call environmentally responsive materials. So you can time the release. You can only release material when it gets hot, or you can only release material when the plant is stressed and making a lot of reactive oxygen on its own. And then ultimately, we have to make these approaches scalable. Because if you think about agriculture, and I love this picture here to the right, the scale of agriculture is so enormous that you always have to have scale in mind when you're thinking about any of these solutions. Because if it's not scalable, then no one's ever going to use it. And of course, in all of our work, we can have various things to control the properties of the materials, the charge, the size, the solubility of the material, the morphology, shape, the coatings that we put on the outside. These are all the knobs that we can turn to tune the materials to get them to do what we want them to do. All right, so let's start with the first part here, which is leaf uptake. Can we put things into the leaf? Because that's pretty much the first step of the process if you're going to foliar or apply something. And the idea is here, you put a particle on the top, on the cuticle. It needs to get through the cuticle and the epidermis and into the mesophyll. And we have this instrument. It's a hyperspectral microscope. Cytoviva makes it. And we can essentially focus the plane. A focus can be on the surface, could be inside, could be on the top of the epidermis, it could be in the mesophyll. So we can sort of focus and then look for our particles. It's kind of like confocal microscopy, has pretty good Z resolution, but it uses a slightly different principle to pick up on the particles themselves. All right, so one of the very first things that we did when we started with the leaf uptake, and this is published some years back, and Astrid sort of led this. And interestingly, Jay was a master's student here. She ended up at MIT too, in the same lab that Elin's going to be going to. So it's kind of a small community, I guess. But we took some particles and we put them on the plants, and we were just playing around with size. We wanted to know, do small ones go in and do big ones not go in? And we wanted to get some understanding of the size cutoffs for uptake into the leaf. And we tried a couple different coatings. We put this PVP coating on, polyvinyl pyrrolidone, and we put a citrate coating on. And you could think of this as a hydrophilic coating and more of a hydrophobic coating, or relatively more hydrophobic coating. And then we just wanted to see where the particles went. And we got a very, very interesting result. So what you see in the top left here, this is just the top of the wheat leaf. So this black kind of splotch here is the gold particles. They aggregate and they sort of look black. This is just an optical image of the top of the leaf. And what you can see is, regardless of size, there's a lot of particles with the citrate coating. This is the citrate coated one. A lot of particles just sort of stuck on top of the leaf. But when we use this PVP coating, this polyvinyl pyrrolidone coating, we went back to that spot. And the biggest particles, the 50 nanometer particles here, a few were kind of stuck maybe on the surface of the leaf, but the rest of them were just gone. They just sort of magically went into that leaf. And this polyvinyl pyrrolidone is actually a pretty magic polymer. It's used for a lot of things, like in eye drops, and it's used in foods and other things. But it does a pretty good job at moving stuff through these wheat leaves. And then, if you take a wheat cross section of the leaf, we saw some interesting things. So this dashed line here is the top of the leaf. So this is really just a cross section through a wheat leaf. This is just some synchrotron X-ray fluorescence mapping. The red is the gold. So the citrate particles kind of were stuck on top of the leaf, just like you see here. So it matched that visual image quite well. And then inside the leaf, this is the vasculature bundle, and outside here is this mesophyll area. You can't see any red. So anything that would get in, and it's hard, you can't tell yet from these pictures that things get in, but they do. Anything that went in kind of disappeared fairly quickly. It got into this vasculature and disappeared. Whereas the PVP coated particles, there's nothing on the top of the leaf, almost nothing, a couple little specks here, but not very much. There's a lot of gold. It's kind of this pinkish color inside here. There's a lot of gold inside the mesophyll and a little bit in the vasculature. So everything went inside, but a lot of it sort of got stuck in the leaf. And some of it does move. I'll show you some of that data in just a minute. But the bottom line here is a different coating, whether it's more hydrophilic citrate or more hydrophobic PVP, made a big difference in terms of how it interacted with the leaf and how it got inside. All right, so we've since moved to these polymeric materials, so star polymers or these bottle brush polymers. We do the same thing, we put them on the leaf, but instead of synchrotron, we're using this Cytoviva instrument here, which is just essentially looking for fluorescence. So we load these with crystal violet so we can find them. And what you can see here, this is the bottle brush, actually, and this is a star polymer. So star polymers are these little spherical ones, and bottle brushes are the long ones. What you can see is on the epidermis, we don't see anything. Literally, there's nothing sitting on top of the epidermal plane here, because everything that we put on the leaf moves inside. It's almost all in the mesophyll, which is shown here in red. And then if you take this leaf cross section, you can see that a bunch of it is near the vasculature, kind of entering the vasculature. If you look at a leaf cross section, which is this bottom one, and you look at the away from the vasculature, it's hard to tell, but you can kind of see that this stuff is distributed throughout the leaf. It looks to be in the apoplastic space, kind of between the cells, but it's hard to tell at this resolution exactly what's going on there. And we see quite similar behaviors for both the stars and the bottle brushes. So here shape isn't playing a big role. The spheres and the rods are behaving quite similarly. But the bottom line is they go inside, they go inside nearly 100%. So there's potential to be highly efficient with this delivery approach. Now, if you do it with a positively charged particle, I guess I should point out, all these bottle brushes are negatively charged, and this is what we see. If we do this with a positively charged bottle brush, we start to see a bunch of material on the epidermis. So this red is showing the material on top of the epidermis, and epidermal cells kind of look like this jigsaw puzzle looking thing. And we have a bunch, but we also have some in the mesophyll. So some does move into the leaves, but a lot more stays on the epidermis. So harder to put positively charged things in than negatively charged things, which makes some sense given that the cells have a slight negative charge to them. Very recently, and this is a lot of this is kind of a mixture of published and unpublished work. We very recently, I had this guy Kurt Ristroff here. Well, he's still here until August. He is going to start a faculty job at Purdue in August. He has a different way of making particles. It's called this flash nano precipitation. It's actually the same thing that's used to make, I think, the Pfizer vaccine. One of the two vaccines. This is the process. So it's highly scalable. We know that. But we made some particles with this process, and we put them on these citrus leaves. So citrus greening is a big problem. It's a phloem disease. So you need to move things into the phloem. Citrus leaves are hard. They're notoriously difficult to move things into because they have this really thick cuticle on them. But it turns out some of these particles we've made actually do go inside, and we can see them. This is inside the mesophyll. With the yellow is where our particles are. So we know we're delivering particles into the mesophyll. At least we don't know about the phloem loading yet. We need to measure all that. But we're kind of moving towards a different platform to make particles to deliver things. But it looks pretty promising there. All right, so we can get things in. Now the question is, can we load the phloem? And for phloem loading, it means you're going to put it on the leaf and you're going to go look for it in other parts of the plant, whether it's the roots or the stem or the older lower leaves or the newer younger leaves, or maybe the fruit. If you take it all the way to fruit, we're going to look where this stuff goes. And again, we want things in phloem because a lot of diseases are in phloem as well. Plus, if you want to load nutrients into fruits, you got to load the phloem to do that too. All right, so this goes back to that first paper that I showed you where we put the gold nanoparticles on the leaves. And I showed you that they went into the leaves, got stuck in the mesophyll. If we go in and we look at where they went in the rest of the plant, this is all color coded. So this gold color, these gold bars here is the percentage or the fraction of the material, I guess it's done as percentage, that goes to the roots. What we found, interestingly, was something like 25% went to the roots. The small ones, 12 nanometers and 3.5 nanometers, actually exuded the roots and ended up in the soil, the rhizosphere soil that was adhering to those roots. So we could put something on the leaf and have it move to the roots and then move outside into the rhizosphere soil, which is pretty cool if you're trying to target nematodes or something like that. Okay, so we know a good fraction of this material is actually moving around plants. And what was interesting to us is even 50 nanometer particles were making their way down to the roots. That was way bigger than we thought. Our 50 nanometer particle was the one we thought wouldn't go anywhere. It was going to be our largest size that was our cutoff, but it turns out that they moved down to the roots just fine. They don't exude into the soil, interestingly. So there is some cutoff between 12 and 50 for that exudation out of the roots. But this was kind of an interesting find. We didn't have, that's just two particle types. We started wanting to play around with how do the properties of these things affect where they go in the plants. So Elin, I guess I should say, Elin made these polymers. So we use this beta cyclodextrin and we do ATRP and we grow a bunch of polymer arms off of a thing. And we end up making a core shell particle with a PAA core, which is negatively charged, and you can load something in it. In this case, we load gadolinium so we can go find it. And then you can have whatever shell that you want. And these star polymers, we made them different sizes. You could see their hydrodynamic diameters range from about 13 nanometers to 30 some odd nanometers. So we're looking at size. We can also look at charge. As I said, we load these things. We load this PAA core with gadolinium, and it stays inside there. It's strongly bound. We've shown that it doesn't come out. So what we do is we put these gadolinium loaded materials on the leaf, and then we go look for gadolinium in the rest of the plant, and it has to be our star polymers. We can figure out where they go. And you just simple ICP-MS to do that. All right, so what have we done here? Elin made a whole series of polymers with different cores and different shells. We looked at something like size. So we have small, medium, and large, and you can kind of see these top three are the sizes. They're fairly monodispersed, and there's good limited overlap between the sizes. All their zeta potentials are pretty similar to each other. So we can look at size. Another set was exactly the same size, but they had different charge contents associated with them. And you did that by changing the core to shell composition, and the core has charge and the shell does not. So The more core you have, the more charge content you have. We played around with hydrophobicity. So this PMSEA and PMTEA are one's hydrophobic, one's hydrophilic. And if you change the different ratios, you can change the hydrophobicities and keep the size and the charge relatively the same. So played around with all of that. And this was published last year. I'm going to give you kind of a summary. So one of the things that we hypothesized was that all of those factors, charge, size, hydrophobicity, would affect phloem loading. And then it turned out that it didn't affect phloem loading at all. All the particles we put on, approximately 30% of what we put onto those leaves ended up in the phloem, regardless of size or charge or hydrophobicity. So that was actually a little disappointing. We thought that we could get some interesting, keep things in the leaves or put them in the phloem, move them to roots, and we thought we could do that. But it turned out that we couldn't affect phloem loading with any of those properties. But if you look very carefully at where the particles go, so phloem loading just meant it went to some other part of the plant. But if you look very carefully at where they go, whether they're in the stem, and all these colors are color coded to this plant here. So the blue is the stem and the orange is the roots. So we can look at this thing and we can say larger size materials kind of preferentially went to the roots relative, and the younger leaves relative to the other parts of the plant. And this is held up, whether it's a polymer or a big particle or a carbon nanotube, whatever it is, bigger things tend to move to the roots better. And then if you look at charge content, overall, things that had more charge were less mobile. Things that had less charge were more mobile. So these unhatched ones have bigger bars in other locations, and they're the ones with the least amount of charge. Okay, so charge does affect whether something moves from the leaves or not. And then hydrophobicity didn't seem to do anything. And again, that was shocking to us, but it might be because all of these particles are pretty hydrophilic because they will disperse in water. The relative hydrophobicities are different, but overall, maybe they're all just a little too hydrophilic, because we know hydrophobicity should affect things, the way things move in plants. That's a well-studied problem with plant protection products. All right, so that was kind of an interesting find. We've since played around with the shape. So all of what I just showed you were these spherical particles. We're now making these polymer bottle brushes that are a little bit longer. And on the left here are these AFM images showing the stars being these tiny little spherical things about 10 nanometers, just about what we think they're supposed to be. These middle-sized ones are kind of these elongated brushes, and then these very long brushes look like these kind of worm-like structures. These were actually done at MIT. Pretty nice AFM they have there to be able to do this. Interestingly, the transport, regardless of whether it was a sphere or a very long bottle brush, was all around the same. We're still around this 30% number of stuff moving to other places, which is telling us that it's this one size, sort of it's the smallest dimension of the material that's controlling how it moves in the plant, at least up to a couple hundred nanometers in length, which is what these longer ones are. And then, as I showed you before, the positive charge ones didn't go through the epidermis quite as well. Well, it turns out the positive charged ones don't translocate quite as well as the negative charged ones. They do translocate, but just not quite as much as the negative charged ones. So we're starting to get some understanding of how these properties affect their translocation in plants. All right, so now that we know how to move things around, the other question we had is, can we make materials that will respond to the environment and deliver agents in response to that? And two things we looked at. One is temperature. So when the plant gets hot, can we release something to manage that stress? And the other was just general ROS. And when a plant gets stressed for any reason, it makes a lot of reactive oxygen. Can we scavenge that oxygen and then release an agent in response to that scavenging and promote plant photosynthesis that way? So I'll just sort of show you a few of these approaches. And both of these are published papers. One, like maybe, what were they, 2020 and 2022. So the ROS responsive material is this PMSEA and PMTEA material. So it's a core shell, And what happens is the chloroplast produces some reactive oxygen. The reactive oxygen reacts with PMSEA, converts it to PMTEA, and it changes the hydrophilicity or hydrophobicity of the polymer, which changes its conformation, which then ultimately releases some of this magnesium out of the core. So think about the plant. The plant's reaction is affecting the material's properties, which is affecting the release of the drug. And it's sort of twofold, right? One is it's scavenging ROS, which we know helps the plant. And then it's releasing magnesium, which we know promotes photosynthesis because it's needed to enhance photosynthesis. So that was the concept. It was a kind of a bold concept. We weren't sure that that was going to work, but we tried it anyway, and it turned out that it actually worked pretty well. This gives you an idea of the synthesis procedure. I won't go through that in all detail, but it similarly starts with this beta cyclodextrin. We grow all these polymers, and we make this diblock polymer that has what we need in terms of the two blocks, the MSEA and MTEA. And then we load them with magnesium. They're negatively charged. When we load it with magnesium, they're a little less negatively charged, which makes sense because we're putting a positive charge into the core and offsetting some of that charge. We also made a control polymer that doesn't scavenge any reactive oxygen, but it's similar in size to the reactive one and charge. So we wanted to compare a reactive one to a non-reactive one. And you can sort of see here, this is the chart. This is the size distribution. They're all quite similar in size. All right, so the first question we had is, do they scavenge ROS and do they release magnesium in response to that? So what you see here on the left is this fluorescence signal. And if you're quenching fluorescence, that means you're scavenging reactive oxygen. And we're just putting hydrogen peroxide in as our reactive oxygen species. So if we have a control, we're making a lot of this fluorescence due to the reaction with the dye. By the H2O2. And if we put in, I guess this guy is a 0.05 grams per liter of a control polymer. So this is our non-reactive one at a low concentration. There is a little bit of scavenging that's going on, but not very much. Here's the same concentration of polymer, but our reactive one. So there's a big drop between the two. And then if we go to a higher concentration, this is our control polymer that is scavenging a little bit of stuff. But this is our reactive polymer at, in this case, 0.5 grams per liter. And you can see that it's pretty much scavenging all of the ROS in the system. So it's doing its job. And in vitro, this is not in the plant yet, although I'll show you in a minute, we have some data for inside the plant. And then this shows the magnesium release over time as a function of pH. The important thing here is the red ones are with the reactive oxygen and the blue ones are in the absence of the reactive oxygen. So clearly when ROS is present, we're getting this reaction and quenching and we're getting the release of the magnesium. So the material seems to be working the way it's supposed to, at least inside of a beaker. And then we do the same thing. We put things on the leaves. We go and we look where it is. And we found some interesting things. Again, nothing on the epidermis. So it's moving into the mesophyll. If you look at the images of the mesophyll, what's interesting about this, the red shows where our particles are, based on the spectrum that we see in each of the pixels. It looks like it's sort of associating with the chloroplast, which is good because that's kind of where we want this stuff. We want this stuff to be near the chloroplast and deliver the magnesium. You know, it's hard to know for sure with this hyperspectral imaging. So what we did is we extracted a bunch of chloroplasts and we looked at how much of the gadolinium in the particles was associated with chloroplasts. And it turned out that about 30% of the gadolinium that we applied ends up being associated with the chloroplasts when we have it in our star polymers. If you just put free gadolinium on, a lot less is associated with the chloroplasts. So we're pretty sure that this stuff is going through, it's getting into mesophyll, it's getting through cell walls and cell membranes and associating itself with chloroplasts, which again is a good thing. And that was somewhat fortuitous. We weren't sure where this stuff was going to go, and it behaves a little differently than some of our other materials, but it's where we want it. And then with Juan Pablo and Suji at UC Riverside, what they do is they use confocal microscopy. And they actually showed that this is now inside of a live plant leaf. I guess it is live. They look at these in a live way. And then this is our control where we're seeing a lot of fluorescence. And then we're seeing that our control polymer is maybe quenching a little bit of fluorescence, but not much. But our reactive polymer is indeed quenching a lot of the reactive oxygen inside of the leaf. So it's behaving the way we intended it to behave in vivo, which was pretty neat. And then we do a bunch of testing with this where we look at the ability of the plant to assimilate carbon dioxide into plant mass, and as a function of either stressing it with CO2 or stressing it with light. And it turns out that our treated plants are in these blue ones are statistically significantly better than the untreated plants under these stressed conditions. So the material is also working, the plant's happier, is pretty successful actually for something that we just kind of randomly designed and we're hoping that it would work. All right, then more recently, we've been playing around with this heat stuff. We did publish something in 2020 on heat activated materials, and we've kind of upped the game a little bit. And now instead of just showing that you can release crystal violet inside a plant when it gets hot, here we're going to load spermidine, which is a plant hormone that can manage stress. So we have these bottle brushes, we load them with this spermidine, we put them inside the leaf, then we're going to heat the leaf up. And when we heat the leaf up, the idea is that the material will change conformation, because this outside is a polynipam layer, which will collapse when it gets above a certain temperature. When it collapses, it releases the spermidine. The spermidine helps the plant overcome its heat stress. Now, what we really wanted to do, the goal here was to put this on a plant and have it kind of stay around inside the leaf for an extended period of time. And then when we heat it up, it would be active and ready as soon as it's needed, right? Because you never quite know when a heat wave is going to come. You don't know which day. And if you had a massive field to treat, you're going to have to put the treatment on the field ahead of time. So you want this material to be there and active and ready to go. All right, so this just shows you kind of some of the very similar data, right? We made these polymers, we load them up. This is spermidine here, this is the molecule. This is the size of the materials that we made. It matches what we think they are. And then the bottom line here again is spermidine release at higher temperatures. 40 degrees C is higher than at 20 degrees C. So we are getting this heat responsive release of the spermidine, which is what we're trying to do. If we do the same thing with our Cytoviva, we put these things onto the leaf. Again, nothing's on the epidermis. A bunch of it's in the mesophyll, which is where we expect it to be. Some of this going into our cross sections, some of this is entering the vasculature as we expect it to do, and then the others is kind of hanging out here in the mesophyll, right around the different cells. And again, this is where we want it, right? When it heats up, we want this thing to release spermidine, then the spermidine is going to be taken up by the cells, and then it's going to help manage the stress inside those cells and promote photosynthesis. And then this is where it got really interesting. Right, so people do use spermidine, and you can just spray spermidine on the leaf, and it will work. It will alleviate stress. So what this shows you, this is 1 day after application. So we sprayed some plants, we sprayed with our bottle brushes with the spermidine. Others we sprayed with just some free spermidine. And what you see is the stressed plants are at the treated plants, which is both the free spermidine and the brushes. They kind of behaved the same. They definitely did better than the control plants. So the treatments helped in both cases here, both light CO2 stress and heat stress. They're helping the plant. But it was a little disappointing that both free spermidine and spermidine in our bottle brush did the same thing, because then what's the point of the bottle brush? It's not doing very much. But what was interesting is we said, okay, well, let's take those same plants and let's go back 15 days later and let's heat stress them again and see what happens. And when you do that, the free spermidine that you sprayed on those plants is no longer active. The plant's not using it. It was probably broken down, or it's washed off, or something, it's disappeared. But the bottle brushes are still releasing 15 days later the spermidine into the plant, and it's managing the stress. So So that's pretty good. You know, if these are your crops, you don't want this to happen. But if you went and you knew it was coming in the next 2 weeks, you sprayed, your plants are protected for an extended period of time, which I think is kind of an interesting concept of inoculating plants against heat stress. All right, so I'll wrap it up and then take any questions that might have come up. So where are we here? We believe that the properties of these polymers and various particles can be engineered to deliver them into plants. We're starting to make some pretty good progress on understanding how charge and size, et cetera, are affecting the mobility and the locations that they transport to. 30% phloem loading is huge. I don't know if you're in the ag world, you know that that's tenfold bigger than what is normally achievable. So that's a good thing. Can we do better? Maybe. We haven't been able to yet, but I think we could work at getting more into the phloem. We know about how large sizes are encouraging things to go to the roots, smaller sizes go to younger leaves, and charge, et cetera. We went through all that. It's in the paper. We know we can start making and are making these responsive, what I call environmentally responsive materials. They respond to ROS, they respond to temperature, and they deliver this cargo when they're inside the plant. So we're kind of moving towards drug delivery in plants, right? And the challenge that you have is if it's cancer drug, you're willing to pay just about anything for it because you'll die if you don't get it. For agricultural materials, people are not willing to pay quite that much. Although there are probably some cases where if you're going to lose your whole field or pay a certain amount to inoculate them once, that maybe you would pay. So I think we don't quite know what the economics of this whole thing is just yet. But the exciting thing is this idea of extended release inside the plant seems to be possible. All right, so where are we headed? We're doing some flash nano precipitation. That was the stuff I showed you with the citrus plants. It's a new way to make particles, well, it's actually not a new way to make particles, but it's a new way for us to make particles that we can deliver into the plants. It gives us a lot of flexibility in terms of what we can put inside the particles. We're looking at biotargeting approaches, so we're starting to put peptides and things onto the particles to get them to go to the chloroplasts or go to certain parts inside the plant. We just got a new big grant to look at delivering nitrogen this way. Can we deliver nitrogen with particles? That's hard. I'm not going to lie. That is proving to be very challenging, but we're working on it. We got 4 years to do it, so I think we'll get there. And then we're also looking at various biopolymers. So I showed you a bunch of synthetic polymers that we make because we can control the properties, but there are there biopolymers, waste products, for example, that are GRAS, which are generally regarded as safe because that makes their adoption easier. Can we start using those for some delivery? All right, and I'll go ahead and stop there. I kind of lost track of time, but hopefully we get some, yeah, we should have some time for questions. And I got to thank the NSF funds just about all of this research at this point. We should get some USDA money here pretty soon, but for now, it's all been NSF. So thanks, and I'll stop.
Thank you so much, Greg. That was fascinating talk, and you're doing some really, really interesting work. So now I would invite the audience to ask any questions that you have. Feel free to open your cameras and your microphones and ask your questions live, or if you prefer, you can put your question into the chat and we'll just read it from there. And maybe while we're waiting for people to organize their questions, I'll ask you one to start, Greg, and that's with these PAA stars or the bottle brushes. You showed us gadolinium and magnesium that you put into the core. I'm just wondering about when you start to think about organic chemicals, pesticides, for example, what sort of issues you run into, particularly in terms of stability within the core, if you're going to start to add organic chemicals into that kind of thing, or would you expect that you might need a different type of polymer to deliver those kinds of chemicals?
Yeah, so it's a great question. Let me go back to, this is part of the reason that we're doing FMP, which is unfortunately way back to, oh, here we go. So this is why we switched to this system, and I didn't go into the details here, but this is, so Kurt was in Bob Perdum's lab at Princeton before coming here. Bob Perdum is basically a drug delivery guy, and they deliver very hydrophobic cancer drugs to people. So this system is actually set up to put hydrophobic chemicals like a lot of the antibiotics and the various pesticides that we want to deliver into the core of these particles. So that's in part why we switched to this FNP process, because it allows us to deliver those kinds of materials. If we just want to deliver metals, we could deliver the kind of like the stuff Jason was talking about. You could deliver copper oxides and copper phosphates, and you could deliver sulfur that way. But if we want to deliver other metals, we have to be a little more clever. And if we want to deliver hydrophobic organics, we got to move to this system here.
Okay, thank you. I see we have a question on the chat from Shanghai Jiao Tong University. Thanks for your speech. I have a question. After the particles are transported into the young leaves and roots, what will then happen in these young leaves and roots?
Yeah, so the ultimate fate of these particles is an important question. And it's one, we've been focused on understanding the chemistries. We've been focused on making these synthetic polymers because we can control their properties. We're trying to figure out the design rules for these systems. Ultimately, when you use them, you're going to have to move to biopolymers. You're going to have to move to biodegradable materials. And I'd imagine that if we can make a biodegradable material, the ultimate fate would be that they're in the leaf, then the plant senesces, goes back into the soil, and degrades along with the plant matter. That's kind of the ultimate goal. The materials that I just showed you are not highly degradable, so those would stay with the plant primarily. And if it's something like maize or wheat where you're not eating the leaves, it's probably not such a big deal. If it's lettuce, people are going to get a little more upset about eating what you've sprayed on those plants. So that was kind of a long answer, but the long-term goal is to create biodegradable versions of these things that can have these properties, but will be harmless and biodegradable in the system.
Maybe I can just come back to your answer to my previous question and the FNP process. Is that scalable? Like how feasible is that to use if we want to start producing chemicals that are going to be distributed globally?
So one of the reasons we're using it is because it is highly scalable. So it's just it's a impinging jet mixer. It is what's being used to make I believe it's the Pfizer COVID vaccine. So they're making a lot of that right now. Now agrochemicals are even larger masses than that. But because it's an impinging jet, you in principle could design it to be as large as you want it to be and produce a lot of this material quickly. And there is one shop in China that Bob Perdum was working with that scaled this up and is producing things at scale with this process.
That's good to know. Other questions? So for Greg. Any other questions for Greg? You've given us a nice also introduction or start into our next week's talk, and I'll just mention that while we're waiting for other questions. But our next week's speaker is going to be Dr. Craig Poland from Edinburgh University, and Craig's going to be speaking on medical, industrial, and environmental uses of nanomaterials and some of the resultant human exposures from these uses. So some of what you've talked about today, Greg, will flow nicely into that talk next week. We have a question from Fei Dong in Nanjing. Thanks for your presentation. You're interested in foliar application of these nanoparticles. So what are the advantages of foliar application relative to a soil application?
Yeah, very good question. So when we first started this, we were working with soil application, and we were thinking about, okay, we'll put things into roots because that's how plants take things up. Well, it turns out that the xylem system, which is the part that takes things from the roots up to the rest of the plant, is not very efficient at moving particles. And in fact, most translocation factors, you might get 1% of what's on the roots that moves up into the higher parts of the plant. Maybe 5% for things like lettuce that are pushing a lot of water and that transpire a lot. So we moved away from that because we were not getting enough into the aerial parts of the plant. So the key advantage, I think, is that, as I've shown, is that we can put these on the leaves, and 100% of what we put onto the leaf can go inside the leaf. So we have the potential for 100% efficiency of uptake and use of the applied nutrient or the applied pesticide. So that's one big advantage. And then the other advantage is that we're loading the phloem with 30% or so of our materials, which is way more than you will ever get with a soil application. And then if you think about soil applications, that's the old way. That's the way that we've been doing it forever. And you don't have much control. You put something in a soil, you hope that a root gets near it. You hope that it doesn't rain really hard and it runs off into a river or into a lake somewhere. So this concept of putting something in the ground and putting an excess amount in the ground in the hope that some fraction will end up inside of your plant is just, I don't think it's the way we ought to be doing fertilizations. I think foliar can be much more efficient if we can overcome these barriers.
Okay, well, I see we've actually hit our 9 o'clock time, and unless I'll give one more chance for any other questions. Yes, there is one more question. So this is from Margaret Graham in Edinburgh. Thank you for your excellent presentation. Well, she'd like to ask about current regulations controlling foliar applications of nanoparticles. Is there an approval process similar to that for other chemicals?
Yeah, that's also a wonderful question. On Friday, I fired off a bunch of emails to the US EPA, to Jim Allwood and some others, trying to understand the process here. So with agrochemicals, you register the active ingredient. So, and it's very expensive to register a new active ingredient, something that's a biocide or antibiotic or something. Once the active ingredient is registered, then reformulating it in a new way, the barriers, regulatory barriers, are much lower. When we start getting into nano, that's when it gets fuzzy. And I'm working to find out from the EPA, and there's also a company called Vive Crop Protection that we work with, and their whole business model, they're in Canada actually, and their whole business model is to reformulate chemicals to make them more efficient, and that makes it easier from a regulatory perspective. So that's kind of a long way of saying I don't know yet, and I think it's a little fuzzy, and I'm trying to find the right people who can help me. But a USDA grant that we're writing right now requires us to include a commercialization plan. So we're trying to answer that question now.
I see that Warren has a question. Go ahead, Warren.
Hello.
Hi.
I just want to say this lecture. I am so thank you for your lecture.
Sure. Thank you.
Thank you.
Okay, thank you very much. Well, I think I think that's everything then. So Thank you once again, Greg, for an excellent talk. I think you can tell by the questions that a lot of interest in this subject area. And as you said, it followed really nicely on our talk last week from Jason White. And from here on, we'll move a little bit more into some of the human health aspects and some of the geochemistry around what happens to nanoparticles once they get into aquatic systems. So I hope that you will join us for our talk next week and the ones following that. And thank you very much, Greg, for helping us today with this lecture.
Great. Yeah, thanks for having me, Doug. Great series.
And thank you all for attending. I hope we'll see you next week. Bye for now.
Take care.